Okay, so we're in the second part of the introductory lecture to evolution as we move along here and finish up evolution. So genetic equilibrium is when we look at allele frequencies at a locus are not changing and the population is not evolving, and the Hardy-Weinberg law of genetic equilibrium comes in. So when equilibrium is reached, allele frequencies are not changing and a population itself is not evolving. Things are staying the same. That's genetic equilibrium. And we've done a lab and we've done some work with the Hardy-Weinberg law of genetic equilibrium. Now, we use the formula for Hardy-Weinberg to determine the allele frequencies of certain traits within our classes. But for those frequencies to remain the same, for the classes to be in genetic equilibrium, there would be 5 conditions that need to be met. So for a species to maintain genetic equilibrium, there would have to be no mutation. Okay, so no new alleles are entering into the population through mutation. There would have to be no mating, or there would have to be no preference in mating amongst the individuals. There would have to be totally random, because if mating is selection based, then there will be a shift towards traits that have higher selective value. So the example we used of birds of paradise, where the birds of paradise have flashy colors and displays, those are used to help females select them. If that happens, if females select based on a preference, then those traits will be favored, genetic equilibrium will be disrupted, and selection will directionally shift towards those traits. Gene doesn't affect survival or reproduction. So basically, it doesn't make them more fit to survive. So the beaks of finches, a larger beak, has to have the same survivorship as a small beak. So it's important that for Hardy-Weinberg, for genetic equilibrium to be maintained, you have to have that. Population has to be large. That one is one of the more easy ones to maintain. Obviously, a large population is something that there are a lot of. And no emigration. So you can't have genes leave the gene pool, because then that changes the frequency and it's no longer in equilibrium. So do you see how for a species to maintain a balance, it's very, very unlikely, okay? Because all of these things are things that maybe you can have 2 or 3 of them, but to have all 5 would be very difficult. So when we look at microevolutionary processes that are going to drive evolution, all right, drive a population away from genetic equilibrium, you have small scale changes brought to frequencies brought about by natural selection. So the nature choosing which traits are best suited to the environment. You also have gene flow, which is the movement of genes between populations and all that, and then genetic drift as populations, because of some event, they're going to move away from equilibrium. So the Hardy-Weinberg law: at genetic equilibrium, proportions of genotypes at a locus with 2 alleles are represented by the equation p squared plus 2pq plus q squared equals 1, which is the formula that has been used by us several times. We used it in genetics. We used it again in the lab, the Hardy-Weinberg lab. And so the frequency of big A is p, little a is q, and we see right here why the Hardy-Weinberg rule. So big A is p squared, big A, little a is pq, and because it's there twice, it happens twice, 2pq, and then q squared. Okay? So obtaining frequencies in gametes, which percentage of each phenotype. So we see right here the population we're able to solve for based off this. So if you look at this breakdown right here, we would normally solve this as we would have the 0.09%, or the 9%, 0.09 or 9% of little a, and then the square root of that would give us our Q value, and we know our Q value here, as we see it, is 0.3, and then we use that to solve for our P value, which is 0.7, and then 0.7 squared would be 0.49, and then you would multiply 0.7 times 0.3 which will give you 0.21, times 2, which will give you 0.42, which is our big A, little a. Now, this is a little bit backwards the way they do it. Obviously, the way we've practiced it works very well. You start with the recessive, which you always know, because the recessive trait is there, and you can find the frequency, and then you go and you base. Once you get Q, then you get P, then you get P squared, then you get 2PQ. This one's just showing that if 0.49% of the population is big A, and 0.9% of the population is little a, and 0.42 is big A, little a, that 0.42 you cut it in half, because 0.21 would be the big A, 0.21 little a, and you add it together, and that's where these values come from. So it's just a little breakdown, but obviously we have our way of doing it to solve it, and that works very well. So obtaining gamete frequencies when given one allele group in a population. In a population of 1,000 people, 90 people have blue eyes. What percentage of the population would have heterozygous for brown eyes? So we do that. We get our 90 divided by 1,000. The whole population gives us Q squared. Square root is Q. Then we get P from that. Then we get 2PQ, and we go from there. So that's exactly what we've always done. We start it that way, and we solve it. Genetic equilibrium. Went one too far. So the starting population, we see that there is no change in allele frequencies amongst this. So no change through generations is genetic equilibrium. So no change through generations is genetic equilibrium. And we see that starting population, generation 2, generation 3, we have the same allele frequencies throughout. So reproductive capacity and competition. All populations have the capacity to increase in number. No population can increase indefinitely. And eventually, the individuals of a population will end up competing for resources. Now we know, once competition comes in, now it's survival of the fittest. Okay? The most fit will survive in a competition environment. So change over time. Over time, the alleles with the most successful phenotypes will increase. We know this. Selection for the most fit individuals. Less successful alleles will become less common. So alleles that don't make any sense don't show up anymore. It's the same thing if a company is making a product. So Apple is making iPhones. The less successful iPhones are phased out. Okay, and we saw that happen with iPods in particular. The less successful ones were phased out until they eventually just had the iPod Touch. Okay, and you just keep the most successful. Least successful decrease, most successful continue. So change leads to increased fitness. So over time, as the selection picks the most fit individuals over and over and over again, it leads to an adaptation of the environment. So now the individuals are best suited to the environment are there. There was a little meme there. Natural selection. Where did it go here? Went too far back. All right, and then we keep going. Sorry, we saw all this. And then we see right here a little meme, the natural selection interference squad. Obviously, it's just meant to be a joke, talking about how in certain scenarios we have people that make terrible decisions and they make out okay. Peppered moths. Prior to the Industrial Revolution, the most common phenotype was light colored. After the Industrial Revolution, dark phenotype became more common. So we see right here, these peppered moths, selection to this darker moth on the right-hand side picture. Just to the left of the white moth, there's a darker colored moth. And the Industrial Revolution selected for moths with better traits, right, to that environment, meaning that the soot covered the trees and made them better adapted. The peppered moths we'll discuss later on because they're a really interesting case study of how something was proven, then disproven, then reproven. So the results of natural selection, 3 possible outcomes. You have a shift in the range of values, you have a stabilization, or you have a disruption. Okay, so when we look at this, here is a directional selection, which is a shift. Allele frequencies favor in one direction. Example, these moths, So we see that this first chart shows the initial population, and you see how it shifts away from the light-colored moths, and by the third generation, we have a shift, and now the average is a darker colored moth. Okay. Also, pesticide resistance. Pesticide kills susceptible insects. Resistant insects survive better and reproduce. If resistance has a heritable basis, it becomes more common in each generation. Same with antibiotic resistance. Antibiotics came into use commonly in the 1940s. Overuse has led to an increase in resistant forms, and most susceptible cells die out and replaced by resistant forms. So, stabilizing selection, we have selection favoring those in the middle, the intermediate forms. So, do you see how the curve by the third generation is pulled in very tightly to the middle? Okay, now disruptive selection, this type has the curve completely disrupted because the intermediate are selected against. And because the intermediate is selected against, now you have the two extremes being favored, the intermediate disappears. All right. So African finches, selection favors birds with very large or very small bills, and we see an example of that disruptive selection. Sickle cell trait, heterozygote advantage. The allele HBS causes sickle cell anemia when heterozygous. Heterozygous are more resistant to malaria than homozygotes. So again, we have a selection for the heterozygote advantage because they now have malarial resistance. And we see that areas where malaria is common is where you see this happen. Gene flow is the physical flow of alleles into a population, tends to keep gene pools of populations smaller, and gene flow will counter differences that result from mutation, natural selection, and genetic drift. So what happens is the alleles come into a population between different populations. So different populations, individuals move back and forth between them and trade, not literally, but traits and alleles come back and forth to maintain some kind of balance. Genetic drift is a random change in allele frequencies brought about by chance. So the effect is most pronounced in small populations. So it's the same as when you look at like a sampling error. Fewer time an event occurs, the greater the variance in the outcome. So genetic drift, you see it when a small section of a population is separated from the main part of the population. And now that leads to what we call bottlenecking and the founder's effect. So it's a severe reduction in population size, kind of like a bottlenecking. If you were pouring marbles out of a bottle, it would select only a few to move through. Causes pronounced drift, may lead to the founder's effect. So this is an example. The elephant seal population hunted down to just 20 individuals, and the rebound to 30,000 individuals. So it's a big bounce back. And electrophoresis revealed that there's now no allele variation at 24 genes because the population was so small. The variation amongst them was limited. Okay, and that bottlenecking was because you reduce population size severely. This happens commonly not just with hunting, but again with populations being separated from the main group. And the founder effect is the effect of drift when a small number of individuals start a new population. By chance, allele frequencies of founders may not be the same, and the effect is pronounced on isolated islands. So we have an example of bottlenecking leading to the founder effect. Do you see these marbles right here? We have traits that are completely lost because of this bottlenecking effect, and that is as the marbles are poured out, we no longer see those yellow marbles, and maybe the blue and the white were equally represented in terms of alleles, but now in the new surviving population we have very few white alleles. Okay, and finally, inbreeding is non-random mating between related individuals, leads to increased homozygosity, can lower fitness when disadvantageous recessive alleles are expressed, and this happens in small secluded populations and happens in dogs quite often. All right.